Alright, off of that subject and on to the different types of RNA. Alright, so there are three different types of RNA that we're going to talk about. I think before we do the RNA, we should just introduce this idea of the central dogma of biology. So central. So a dogma is a belief system that doesn't change, right? A dogma, it's like if you live your life by a code, you live your life by a dogma. So if you follow every single teaching that a religion says, then you live by that religion's dogma. So the central dogma of bio says that we go from DNA to RNA to proteins. This process of going from DNA to RNA is called transcription. And this process of going from an RNA into a functional protein is called translation. Alright, let's see how this works. To get from DNA to RNA, we're going to do transcription, and the specific type of RNA that we're going to form is mRNA. That stands for messenger RNA. So mRNA, messenger RNA, let me do it for color. Messenger RNA, this is going to determine our primary structure of our protein. If you remember the primary structure of a protein, primary structure of protein is just the linear sequence of the amino acids. So if it's determining the primary structure of this protein, then somehow the mRNA codes for what amino acids are going to be within this protein. tRNA is what we call transfer RNA. I'll show you what it looks like. So let's loop. Okay, so it kind of looks like a cross thing, and it has this site either here or here. I'm not exactly sure if it's the top or bottom, but anyways, it can carry what we call an amino acid. So amino acids are these monomers of proteins, and we need to link these amino acids together to get a functional protein. So somehow we have mRNA, and mRNA kind of gives us this script or this code that we have to work over. Then as mRNA works with rRNA or interacts with rRNA, tRNA can bring this amino acid to the rRNA, and using this mRNA will help bring the right one to get our functional protein. Let's break it down to a few more steps. But let's give one more definition for each. mRNA, messenger RNA, is determining primary structure of protein because of its code. rRNA, it's what the ribosomes are made of. So rRNA make up these subunits of the ribosome. These are the site of protein synthesis. And then we have tRNA. This is a carrier protein that's bringing amino acids to the mRNA as it's interacting with the rRNA or the ribosome so that the amino acids can be synthesized together to form a protein. So this first idea is RNA, or this transcription, which is the conversion of DNA to mRNA. Let's see how it works. When we talked about DNA replication, we talked about DNA unwinding, so let's draw some unwound DNA. We have a 5' end, and a 3' end, 3' end, and a 5' end. So when we're building mRNA, what we're going to do is build again in a 5' to 3' direction. So DNA is going to unwind. Now instead of a DNA polymerase, we're going to use an RNA polymerase. So let's just say on my DNA strand, and I'll do this top one actually. Let's say I have A, I've got G, I've got T, I've got A, and C. Now, my DNA polymerase is going to help bring in these nucleotides to help match with that, but I'm going in a 5' to 3' direction. So now whenever I draw my DNA polymerase, it's going to bring a G here, it's going to bring a T here, an A there, a C there, and a T there. So these are complementary base pair rules, and now I have, it looks like this, and this is my mRNA. So let's take my mRNA. Now I have mRNA. This can also be called a transcript. If you remember, a transcript is the recording of your grades. Well, this is the recording of the information that you want to be to form into a protein. So the DNA can't go straight to a protein. We have to have this intermediate transcript called an mRNA. mRNA is going to lead through the nuclear pores. So if I draw, there's my cell and here's my nucleus. If you remember, we had all these small, those aren't holes, those are dots. We had these small holes called nuclear pores. This is what mRNA starts to lead through. So now we're starting to see, man, I'm using information from that cell lecture, and I'm re-seeing it again to understand how mRNA is leaving the nucleus. So mRNA is going to leave the nucleus, and now we have mRNA interacting with a ribosome. And this can either be a free ribosome or attached ribosome, again, connecting some of those cell structures that we've covered before. And let's see what the mRNA does next. So transcription, mRNA is created from a DNA template. DNA unwinds. RNA polymerase, not DNA polymerase, uses complementary base pair rules to add the correct nucleotides, as we've shown, and then eventually the RNA polymerase is going to arrive at a termination site in this DNA where it's going to say end, and that's where it'll end. So as we're going through this, let me draw a strand of DNA. As we're going through this, let's say our gene is this long. There's going to be different parts that are important. So here the first part we'll have is a promoter. So this promoter is basically saying, hey, this is where you should start making that mRNA. So it doesn't code for anything of the protein. It's just a sequence of nucleotides that says, hey, you're in the right spot. Then we'll start doing some coding stuff. This coding stuff that's going to be important for our gene later on is called an exon. This is a non-coding region called an intron, and we'll just navigate those back and forth. This is my DNA, remember. So now my RNA polymerase can come to the promoter region. Remember, this is called a promoter or a promoter region. And RNA polymerase is just going to start slapping down nucleotides in the 5' to 3' direction. So now we have our RNA transcript, our mRNA transcript, and it's going to have coding parts and non-coding parts. So exons and introns alternating. And these sizes aren't realistic for everything. Maybe an exon is that big, maybe the whole intron's that big, and just this and this are the exon, who knows. Just trying to give some visualization of what's going on. So now we need to we only want this. This is our finished product. So how do we only get the red? We do something called splicing. So we'll make a cut here, we'll make a cut here, and here. And we'll just do something else with these introns. And then we can ligate those ends together, and then we get this nice mRNA transcript. So an intron, it's non-coding segment of DNA, that's that yellow stuff. The genetic information, it's removed from mRNA before it leaves the nucleus to go to the ribosome. Exons are coding segments of this DNA, and they link together to make what we call a mature transcript. And this mature transcript is what we can use. It's going to leave leave the nucleus and go to a ribosome. To be translated. So now, if you think, the best way to think about transcription versus translate, to transcribe something means you're writing it down, right? So if someone's maybe saying something to you and you're writing it down. You're still using words, so they have a certain language, you have a certain language. Maybe it's English, so we're all using the same alphabet and stuff, right? So if you think of DNA and RNA, they both use nucleotides, so they're speaking the same language. So you're transcribing from DNA to RNA because you still have nucleotides. Now that we're going from nucleotides to amino acids, we have to be translating. So we're almost using a different language. So we're going from nucleotides to amino acids to form this protein. So that's how you can remember transcription versus translation. So we're going to come up to a ribosome. Here's a ribosome. Our mRNA is going to come from over here, and it's going to move in that direction, start getting red. So the first thing that's going to be red is here, then this will be red, then this and this. So this mRNA is going to be red until it approaches the start codon, which stands for AUG. Or I should say AUG starts for this codes for the start codon. AUG 99+% of the time is going to be your start codon, and AUG brings the amino acid methionine to the ribosome. So really if I, so I'd really have my mRNA, and somewhere like here I'd have AUG. So it's going to start getting fed this way, and as it gets moved down, the ribosome is going to say, okay, I've started AUG. So now we have to work in these triplets. We call them triplets in DNA. We call them codons for mRNA. So if I look at my mRNA, and I have this, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. A, G, T, A, T, A, U, G, A, T, C, G. So my mRNA is being fed this way into my ribosome, and it says, okay, A, G, T, none of that matters. A, U, G. Oh, boom, right there. Let's go back. A, U, G, this is important. So we're going to have a tRNA. tRNA is going to bring our amino acid methionine. I'm just going to put MET right there, and then we'll show how methionine interacts with other amino acids. But that's the basic idea of translation. The mRNA transcript is going to be fed through this ribosome. The ribosome is going to eventually reach the start codon called AUG, and that codes for methionine. So methionines are almost always your first amino acid in a protein. And these three-letter phrases or words are called codons. And codons, this three-letter is always going to match or always going to be used for methionine. ATC is always used for another one. And then we're going to keep going along, and then eventually we want to stop this. So let's say this thing was 2,000 base pairs, and we just... Hold on, let me clean this up for you all. So we've already fed like 1,000 base pairs, or another name for base pairs, nucleotides. So 1,000 base pairs in. We still have our ribosome and our ribosome, but we have all this stuff. AUG is way down there, right? Well, we're eventually going to keep coming with this mRNA, and we can have one of 3 codons that will stop this thing. So the first one he likes you to remember is UGA. We don't go to Georgia because that's going to stop translation, all right? So UGA is one of them. Another one is UAG. So just switch those letters, okay? The other one is UAA. So UAA. These are your 3 stop codons. This is saying do not add any more amino acids. Your protein synthesis is finished. Methionine start codon, AUG, UGA, UAA, UAG, those are all stop codons. They don't code for an amino acid. All right, I'll read my notes one more time, and that's about it. So, translation, mRNA arrives at the ribosome. The mRNA, we're just going to say it associates with the ribosome, so it kind of attaches, whatever you want to say. And then as the mRNA passes through the ribosome, these codons are read as an READ, like you're reading them. So the genetic code is made up of those nucleotides or base pairs, and they're read 3 at a time, and as they're read, the appropriate tRNA will bring the appropriate tRNA will bring the appropriate amino acid to the ribosome, and we can grow this chain of amino acids.